Good. All right. It is 10 o'clock. We'll get started. Thanks for everyone joining our webinar on Cell Types 101. This is a series on cell types taxonomies. I'm Laurel Filer. I'm an Education and Engagement Specialist here at Allen Institute, and I'll be your moderator for today. So just some housekeeping reminder, this webinar is going to be recorded. The recording will be on the YouTube after this webinar concluded. If you are interested in any future webinar, please make sure you register, as you need a link for all the Zoom and YouTube. We're happy to take questions, post them either on Zoom or YouTube. If you are watching a recording of this, there'll be recording. You can ask your questions in our community forum, which is in our YouTube description. And so just a quick recap about the Allen Institute. We are a 20-year-old nonprofit research company. Our focus is on team science, multiple teams working towards a common goal, allowing us to do some big science, big data, and all our science and data is available publicly on brainmap.org. We also have more than just a reference list. This data includes cell types, humans. Oh, sorry. Sorry, 1 second, technical issues. Oh, sorry. Apologies on that. Technical issues on that. So we also have more than a reference atlas. We have a lot of data available on brainmap.org, including Connectivity Atlas, Cell Explorer, Alzheimer's work, and Brain Cell Atlas. In addition, we have more than our Brain Science Institute. We have including our cell science, immunology, and neural dynamics. But today we'll be talking about cell types. And with that, I'll take it over to Rachel. Rachel, you're muted, by the way.
Hi, everyone. I'm Rachel Hostetler, and today I will be talking to you all about cell types.
Rachel, you're muted. Yep.
Sorry, technical issues. What are cell types? So cell types can be defined as groups of cells with more similar quantitative cellular properties to each other than to other cells. And cells in the same cell type share features defined on one or more of measurable criteria. However, we currently lack standard definitions of cell types within the brain. And so some of the shared features that cell types have include anatomy, morphology, electrophysiology, and connectivity, which then inform their spiking activity and overall their function. And cell types are a product of evolution. They're a product of their genetics, their development, and their environment. And how were cell types historically defined? As I mentioned in the previous slide, morphology has been a really common way to define cell types over the last 100 years. And this is typically done through staining or visualizing cells and then tracing their shapes. Here's an example of some morphological tracings from Ramon y Cajal from over 100 years ago. And by tracing the shapes of the neurons and by analyzing the shapes of the soma, the dendrites, and the axons, researchers were able to see that there were common characteristics shared between different cells based on their shapes. And so because of these shared characteristics, researchers started naming cells based on these shapes. So for example, pyramidal cells have a pyramid soma. Chandelier cells somewhat look like a chandelier. Martinotti cells are named because they have axons in layer 1. Basket cells are named for their shape, and bushy cells are named for their morphological shapes as well. Not only does the shape of the cell matter, but how the cell connects to other cells is also very important. And so cell types were historically defined through their connectivity and by staining and visualizing cells and tracing their connections. And so in the previous slide, I mentioned chandelier cells and basket cells as different examples of different morphologies of cells. And so not only do these cells differ, again, in their shapes, but how they connect to other cells. So chandelier cells are unique in that they specifically target axon initial segments, whereas basket cells target cell bodies, which means they have different functions and different properties within circuits. So the connections that I just described are more local connections, but we can also define cell types by their long-range projections. And these experiments were typically done using different dyes, such as fast blue or dye I for anterior grade and retrograde tracing. And in these experiments, you could inject or deposit dye in one area of the brain and see where that neuron connects to. And due to these experiments studying long-range projections, scientists came up with different names for these types of cells. And for an example here, we have IT cells, PT cells, and CT cells. These are all names for excitatory pyramidal cells, but they differ in where they project to. So IT cells are intratelencephalic, meaning that they keep their axonal projections within the telencephalon. For example, IT cells in the primary somatosensory cortex have connections to secondary somatosensory cortex and also primary motor. PT-type cells specifically project to the pyramidal tract, so they project down to the spinal cord. CT-type cells are cortical-thalamic, meaning that their cell bodies are within the cortex and they send projections down to the thalamus. And so all of those descriptions that I just described are really anatomical and looking at the shapes of the cells and how they connect to other cells. But cells also have unique intrinsic characteristics as well. And one of the ways that we can measure intrinsic characteristics of cells is through electrophysiology. And with electrophysiology, we can use patch clamp to depolarize cells and record their specific action potentials and their firing patterns. And to the untrained eye here, if you're not an electrophysiologist, you can see that these patterns here all look very different between the fast spiking cells, the adapting cells, the irregular spiking cells. And so just by glancing by eye and looking at these cells, you can see that they have different patterns. And so because of this, electrophysiologists started naming cells and defining cells just based on their firing patterns. And so examples of these include fast spiking, adapting, and bursting. Another way that cells were historically defined was through the identification of different molecular markers, primarily by using immunohistochemistry to label different proteins. An example of this are the number of different proteins that were used to identify subpopulations of inhibitory interneurons. So inhibitory cells all contain GABA, but they also contain other really important markers of interest. So a number of inhibitory cells also contain parvalbumin, which is a calcium binding protein. Other inhibitory cells contain somatostatin, which is a neuropeptide. Others contain vasoactive intestinal peptide, which is another neuropeptide. And so by doing different immunohistochemical experiments and double and triple immunohistochemical staining, researchers were able to see that these GABAergic cells have different expressions of different proteins. And so then researchers started calling cells SST or SOM cells based on their expression of somatostatin, or calling PV cells PV because of their expression of parvalbumin. And so I just mentioned a couple of different ways that cell types have been defined, but how do we connect them all together? How can we put together our morphological data, our anatomical data with our electrophysiological data? This is a question that many scientists have been trying to answer for many, many decades. So in this chart here on the left, we see PV cells, which means that these cells contain the calcium binding protein parvalbumin. But we also see that they have differences in the morphology as well. We have both basket cells and chandelier cells in the PV population. And these cells also have a fast spiking electrophysiological firing pattern. And we also know that they vary in other ways, such as their axonal projections and their molecular expression of other proteins. When we look in the middle, we see our SST cells, which express somatostatin, and they also vary in their morphological characteristics as well. We have Martinati cells, which have axons in layer 1. We have X94 cells, which have axons in layer 4. And then we also have different firing patterns as well within SST. And our third group, the 5-HT3AR, the serotonin receptor cells, also vary in their expression of different markers and different firing patterns as well. And so here's an updated chart a few years later from this same lab, adding on to the way that we define these three different types of inhibitory interneurons. Again, in the last few years, you can see this chart has expanded. We have more properties regarding the biophysical properties, synaptic properties, and other anatomical properties. So trying to combine all these different modalities has really been something that many, many researchers in different labs have been focusing on for a very long time. And even more recently, within the last year, there's been another marker, molecular marker that was identified specifically for this other group of cells called the ID2 cells. So again, this is a work in progress, and researchers are continuously finding new ways to define different types of cells. Here's an example of what combining these different modalities looks like in our excitatory cells. So here we have ET cells, which are extratelencephalic cells, which include the PT type that I discussed earlier, and our IT cells are intratelencephalic. So we know that these cells have different long-range projections, but now we also know that they have different genetic markers as well, which is useful for developing mouse lines for isolating different types of excitatory cells. And so why are we studying all these different cell types and further splitting them up into smaller and smaller groups? Well, it turns out that they have very different distinct functions and important tasks such as motor planning and sensory processing. When we look in the motor cortex, again, we have both PT and IT excitatory cells. Again, they have different long-range projections, but they also have different functions and are involved in different aspects of motor planning. For instance, our IT cells are not lateralized for movement, and the PT cells are actually involved in contralateral movement and also movement preparation. In our visual cortex, when we compare PT, IT, and CT cells together, again, they're involved in different aspects of visual perception and sensory processing. So the PT cells are involved in more broad orientation tuning, whereas IT cells are involved in higher resolution visual perception. And then our CT cells are involved in gain control during visual processing. We also see differences in the auditory cortex. Specifically, layer 5 PT cells are involved in learning-induced plasticity, and other infragranular cells in the auditory cortex are actually involved in processing visual information. And so in the previous slide, I discussed motor and sensory processing, but we also know that we have different cell types involved in complex cognitive tasks. So in this figure that I'll be describing, this is a working memory spatial task that was performed by mice. And this is considered a complex cognitive task because mice were trained to go through a maze. They had to remember the pathway which they took, and then they had to repeat through the maze again by taking the opposite pathway. So they had to hold that previous memory in their working memory while executing a new path. And during this experiment, microdrive arrays were implanted over the medial prefrontal cortex of mice to measure the activity specifically of different types of inhibitory cells in the medial prefrontal cortex. And so in this study, they specifically focused on parvalbumin or PV cells and somatostatin or SST cells. And as we click through the different stages of the experiment, so these are different parts of the maze. So again, the mouse has to go through the maze, remember the pathway it took, and then repeat the maze, but by going the opposite direction. And during these different stages of the task, you can see that we have different levels of activity between our PV cells and our SST cells. And when we look closer at the SST cells, we can actually see that there were differences within the larger SST population. There were actually two different groups of SST cells that had different firing patterns throughout this task. And this is important to highlight because if you were to group all SST cells together as one homogeneous group, you would miss this heterogeneity, and you would miss that we have these two different populations that have different activities and firing patterns during this cognitive task. We also know that cell types are differentially affected during disease. So in this figure here, we're looking at differences in neuronal and non-neuronal cells taken from progression of Alzheimer's disease. And this is data coming from postmortem tissue from patients who were diagnosed with Alzheimer's. And down at the bottom, we have a pseudo-progression score. And so as we can see in our neuronal population, all of our cells were declining throughout the progression of Alzheimer's, but they were declining at very different rates. When we look at our non-neuronal population, We actually have the opposite happening for our microglia and our astrocytes. We actually have increases. And again, if you were to study all inhibitory cells or all glial cells together, you would miss these differences in how these different cell types are affected during Alzheimer's. So how do we define cell types today? So all the previous slides that I had discussed was really summarizing many decades of research looking at anatomy, electrophysiology, and connectivity. But today we have newer techniques and newer ways to study cells. And right now, transcriptomics is used to define cell types. And by studying transcriptomics, what we're doing is we're actually studying the RNA expression in the cell. So the genome is referring to the DNA, whereas the transcriptome is referring to the RNA. And by studying transcriptomics and the RNA expression in our cells, we are able to define cell types specifically based on gene expression. Why are we doing this? Well, it's very high throughput. In these experiments, we can analyze millions of cells. We can also analyze many different features of cells. So in the previous immunohistochemistry experiments that I showed you, there were maybe 4 or 5 different molecular markers that were used. And through immunohistochemistry, you're limited by the types of antibodies you have, the species of the antibodies, the different fluorophores that you're using. But with transcriptomics, you can analyze over 30,000 genes at the same time. You can then also compare your results directly to the genome and get better analysis for studying disease and generating genetic tools. Transcriptomics also works well with postmortem tissue, which is really important for a lot of our human studies. It's also universal across organs and species, which is great for our cross-species analyses. And there are also many, many tools that have been generated to analyze this type of data. And now that we can define cell types based on their RNA expression, based on their transcriptome, we can then map those previous modalities that I had discussed onto these transcriptomic-based definitions to really get a better picture of what these cells look like and have better definitions for them. And so what are some basic methods for transcriptomics? So the first 2 methods that I'll talk about are bulk and single cell transcriptomics. And as you can see from this image here, in bulk, all of our cells, we lose the identity of our individual cells, just like we lose the identity of the fruits in our smoothie. They all get mixed in together. In single cell transcriptomics, we can actually maintain the identity of individual single cells, just like our fruit in the fruit basket. And here in this example, again, we can see that if we have our tissue taken for bulk RNA sequencing, again, what you end up getting is the average value of gene expression because all of the cells are being mixed together. Whereas with single cell RNA sequencing, we can actually measure the individual gene expressions of individual cells and then organize those cells based on their genetic similarity. And so what are some approaches to single cell or single nucleus RNA sequencing that the Allen Institute uses? So in some of our experiments, we dissect different regions of the brain, both in mice and human. Sometimes, depending on the experiment, individual layers within the cortex are actually dissected. So the first step is to dissect the region that you're interested in from your sample. Then the next step is to select the cells or nuclei of interest that you would like to study. So for instance, if you're using a mouse line, for instance, if you're interested in studying PV cells and you're using PV transgenic mice that have TD tomato, you can use that fluorescent reporter, that TD tomato, to select for that specific population of interest. If you're working from tissue from humans that doesn't have fluorescent reporters, you can stain with NuN or use other nuclear antibodies to specifically enrich and study those neuronal cells that you're interested in. After you've selected your cells of interest, the next step is to then sequence them and isolate them. The next step is to isolate and sequence those cells. And so there are 2 methods described here for doing the sequencing, SMART-seq and 10x. These are just different ways to do single cell RNA sequencing with different technologies and different protocols. But the end result is that you end up getting the number of RNA counts per cell for your individual single cells. And so by using transcriptomics and single-cell RNA sequencing, we can start to define our cells based on our gene expression. And so after we've collected our cells or our nuclei and we've sequenced them individually, the next step then is to perform supervised unclustering to cluster these cells based on their genetic similarities. So what's shown here is a UMAP, and a UMAP is a dimensionality reduction tool, which turns high dimension data into a nice two-dimensional view. And so the way the UMAP works is cells, each individual dot represents a cell, and dots that are closer together are genetically more similar, and dots that are farther away from each other are genetically less similar. So all of the cells that are colored light green in the center are genetically similar and are different from the cells next to them that are colored dark green or red. Then the next step, after clustering all of our different cells together, the next step is to then organize them based on hierarchy. So we know that different cells are connected to each other in different ways, and that not every group of cells is equally related to every other group of cells. They're actually organized in a hierarchy. After we've organized all of our clusters, we can then start to annotate them based on metadata, based on the gene expression, and research and prior knowledge. And here we can see a dendrogram, which has which displays the ways that all of our cell types are related to each other. So at the top, we have neuronal cells on the left, we have non-neuronal cells on the right. When we follow our neuronal cells down further, we see that they are split into then inhibitory and excitatory cells. And our inhibitory cells, then they are split into cells that are born in the caudal ganglionic eminence versus the medial ganglionic eminence. And then further, we are split further and further into different cell types. And by doing all this clustering and organization and annotating with metadata, then finding a way to store this in a standard format is how we create a cell type taxonomy. And as I mentioned in the previous slides, our taxonomy and our transcriptomics-based cells, we can then compare that to previous data and other experiments based on anatomy, morphology, electrophysiology, and connectivity, because these modalities are very important and give us greater insight into what these cells are doing and how they function in the brain. And so, as I mentioned previously, morphology, again, looks at the shape of the cells. And here we see 3 different cells. And right now, just by glancing by eye, we can see that one has an apical dendrite, the other 2 do not have apical dendrites. They have different branching in their axons, they have different branching in their dendrites, and they have different shapes and sizes. If we also do recording of these cells and record their firing patterns, we start to have a better picture of the identity of these cells. Who are these cells? So the cell on the left, based on its electrophysiology and its morphology, scientists can say, okay, that's very likely a pyramidal cell. The cell in the middle, again, based on morphology and its firing pattern, looks like a fast spiking basket cell. Then the cell on the right, based on the morphology and firing, it looks like a Martinani cell. But we don't know the gene expression of these 3 different cells. We don't know their molecular markers. We can infer, based on previous research, that the Martinani cell likely expresses somatostatin, the basket cell likely expresses parvalbumin, and the pyramidal cell likely expresses RORB, CUX2, and some other markers. But how do we connect all 3 of these together? The next step, this answer can be solved through patch-seq. And so patch-seq is a technique where you can do whole cell recordings to measure and record the firing pattern of different cells, fill the cell with biocytins so that you can reconstruct it for morphological analysis. But then after this process, you can also extract the nucleus and then analyze it for single-cell RNA sequencing. And so here's a video of what it actually looks like to extract the nucleus from one of these cells. And so by combining electrophysiology, morphology, and transcriptomic analysis, we get a much more complete cell. We get a much more complete idea of who these cells are, what their identity is, and what their functions are. So after extracting the nucleus, again, we can do single cell analysis and then measure the gene expression in that individual cell. And we know the identity and the shape of the cell and that firing pattern. So we really have a complete picture of who the cell is and what its function is. Again, by combining electrophysiology, transcriptomics, and morphology, we have a much more complete picture of different types of cells, and we can define them based on the combination of the electrophysiology, transcriptomics, and morphology. So initially, we have 10 cells here in this brain slice. We see their morphology, we see their different firing patterns. And then after extracting the nucleus from these cells and doing RNA sequencing on them, we now can link their genetic expression with the morphology and the firing patterns. And here we can see that the colors matched up with the previous genetic expression. And so this is a further way to analyze these cells better. So I just described patch-seq, and patch-seq can also be compared with electron microscopy. So here in this figure here, in A, we have reconstructions from neurons from a cubic millimeter of tissue that was imaged using serial electron microscopy. And with electron microscopy, you're able to see the ultrastructure of cells. You're able to see all the connections that it makes and every fine detail that you typically cannot see with other types of morphological analysis. And then the great thing about EM is that you can analyze and compare those anatomical features and compare them to data that we have from our patch-seq experiments and match up and say, okay, if this cell has this anatomical features in the EM data set, it likely corresponds to patch-seq data, which then contains information about the transcriptome and the firing patterns in addition to information about the anatomy. And so by combining both patch-seq and EM, we end up getting a much more fuller picture of these different cell types. So in this example, this is an SST or a somatostatin cell. And this is data from, this was reconstructed from serial electron microscopy imaging. And by comparing it to our patch-seq data and comparing those different morphological aspects, we were able to identify that yes, this is a somatostatin cell, because you would not normally be able to find that information just from electron microscopy. So we know it's a somatostatin cell. We know that it's a specific type of somatostatin cell. It's specifically type 8 based on the analysis. And with electron microscopy, we're able to see and measure all the different inputs and outputs that the cell is making. And so this type 8 cell, the majority of their synapses are actually on layer 4 cells. Whereas we look at the somatostatin MET type 4 cell, the majority of their synapses are actually on layer 2-3 cells. And then we have a third example here, MET type 6 again. So these are all different types of cells within the somatostatin subclass, but by looking at our EM data, we can see that not only do they vary in their electrophysiology and their transcriptomic profiles, they also vary in their postsynaptic targets. So we can see that they target different cellular populations and that they target these populations at different rates and also in different layers of the cortex. And so I just wanted to highlight the ways that we can map our transcriptomic data onto these other types of modalities and other types of data that we have. And when we map our transcriptomics with our anatomy and our electrophysiological data, we get a much more complete picture of these cells and their functions. And so a lot of this multimodal research was done through the BICCN, or the Brain Initiative Cell Census Network, to create a mini atlas of cells specifically within the primary motor cortex. And in this project, there were samples from 3 different species, from human, non-human primate, and also mouse. And in this project with BICCN, a number of different techniques, patch-seq, spatial transcriptomics, single cell and single nucleus RNA-seq, single cell morphology, transgenic mouse lines, were all used together to do a comprehensive characterization of cell types within the primary motor cortex. And over 3 million cells were profiled with all these different modalities and across our 3 species. And this ended up generating over 200 terabytes of data which is a very large amount of data to sort through by yourself. And so this was published in 17 different studies, and the project was summarized in a flagship paper in Nature. And because 200 terabytes of data was generated through this project, the Cell Type Knowledge Explorer was created, which is an online tool to help you explore this data and search through it more easily and to make it more accessible. And so the Cell Type Knowledge Explorer is an interactive web application for looking at this multimodal data from the primary motor cortex. And we'll be focusing more on the Cell Type Knowledge Explorer in 2 of our later webinars, specifically on February 28th and March 27th. But for the Cell Type Knowledge Explorer, we took all those 200 terabytes of data generated from that project, and together the Allen Institute with their collaborators created this online tool for you to sort through the data yourself. All right, so back to transcriptomics. I previously talked about bulk transcriptomics, where all of our cells get mixed together and we lose identity. Then I talked about single-celled transcriptomics, where we get to maintain the identity of individual cells and measure their individual RNA expression. And the third type of transcriptomics I'm going to talk about today is spatial transcriptomics, where not only do we maintain the individual genetic identities of the cells, but we also maintain their spatial location within a tissue sample. And why is this important? Well, in the single-cell transcriptomics, what happens is you dissect a certain area of brain, but you don't know where those cells are in that specific tissue. And we know that it's really important to study where cells are located within your tissue sections, because cells in different areas have specific connectivities. So in the hippocampus, also in the neocortex, and in many other areas of the brain, we have different layers and different subregions within that area. And these areas have different inputs and outputs, so they have different connectivities. So it's really important to map out the genetic identity of cells, but also where they are located within your tissue sample. We also know that not every cell in the neocortex is affected similarly by disease. So if we go back to our postmortem data from our Alzheimer's patients, it turns out that different types of cells in different layers of the cortex are affected differentially in disease. What we've seen in our data so far is that cells in the superficial layer, so in layer 2, 3, are more affected compared to cells in the infragranular layers. So again, if you were just to study the neocortex as one area, you would miss that specificity within the different layers and how they're affected by disease. Spatial transcriptomics is also important to study because even if cells have the same molecular marker, they might have different functions during behavioral tasks based on their location within a brain section. So in this example here, this is data taken from a behavioral task where mice were learned to perform a whisker-cued associative learning task. And on the first day, mice moved their heads a lot. And as they learned to perform this task, they stopped moving their heads. So in blue here, we see a normal trace of what it looks like when a mouse learns to do this task correctly. However, in the next step of experiments, they then inhibited SST cells, but only in layer 4. And specifically, when these layer 4 SST cells were inhibited, the mice were no longer able to learn this task. The experiments were also repeated looking at cells in layer 2, 3. And in these next set of experiments, turns out when you inhibit layer 2, 3 cells, it doesn't affect the experiment. Mice are still able to learn this task. However, when you inhibit layer 4, the mice can no longer perform this task. So even though these cells are both, even though these cell types are both somatostatin cell types, so they're within the same subclass, they had different functions based on just their spatial location. So our layer 2, 3 cells and our layer 4 cells have very different behaviors and are involved in different aspects of sensory learning. Another reason to study spatial transcriptomics, well, when we do these types of experiments, we're able to reveal the cell type specific spatial organizations within different layers of the cortex. we're also able to look at spatial relationships between different cell types. So in this figure here, we have 5 different layer 2-3 IT cells, and these 5 different cells are labeled, they're color-coded in orange. And what we see is that the light orange layer 2-3 cells are really clustered together more superficially than our other cell types, so our darker colored layer 2-3 IT cell types. And you would miss this if you just did single cell transcriptomics and isolated layer 2-3. Again, you wouldn't know the spatial organization of these cells and where they are in relation to one another. And this is also important to highlight, especially if these are inhibitory, especially when looking at, cells, particular inhibitory cells or interneurons that make local projections to see which cells are near each other. And so the Allen Institute, together with collaborators through the Brain Initiative Cell Atlas Network, or BICAN, then performed experiments combining single-cell transcriptomics with spatial transcriptomics. And so in these experiments, brain tissue was taken from the whole mouse brain, and single-cell transcriptomics was done to profile 4 million individual cells. Then based on the results from these experiments, then spatial transcriptomics was done with a 500 gene panel, which would then show us the spatial localization of these individual cells that were identified in the previous experiments. And together, this shows us where these cells are located within the brain, and we can look at their hierarchical information. So we can see how they're related to each other, whether or not they're genetically distant or close, by mapping these out through a hierarchical cell type taxonomy. And by doing this, we're able to see our top level of cells or our neighborhood. Then our next level is our class. So excitatory cells, inhibitory cells. Our next layer is our subclass, where we can see different types of excitatory cells or different types of inhibitory cells. Then we have supertypes, which is a more detailed level. And then finally, our last level is clusters, where we can see individual cell types. And then we can use the hierarchical taxonomy to see how they're related to other types of cells. And so this experiment was done again through the whole mouse brain. So the previous experiment that I had talked about, the large scale atlas, focused just on the primary motor cortex, where this was an experiment that covered the entire mouse brain. So the telencephalon, diencephalon, midbrain, hindbrain, and cerebellum. Then after doing these experiments, over 4 million cells again were used for the single cell RNA sequencing. Genetic clusters were defined based on genetic similarity. Then spatial transcriptomics was done to look at the spatial localization. And then at the end, a taxonomy was created and annotated to represent the entire mouse brain. And here's what that actually looks like. So here is the dendrogram showing all of the cells profiled in this atlas here. This is divided again through hierarchy. So we have our cells in isocortex in one area. We have our cells in cerebellum in another area. We have our non-neuronal cells, our neuronal cells. And as you go further through the layers of the dendrogram, you can see in finer detail different classes, subclasses, supertypes, and then clusters. And overall, this represents a whole brain taxonomy containing 4 million single cells that had their transcriptomes profiled, over 4 million cells specifically used for spatial transcriptomics. And overall, this resulted in creating a whole brain taxonomy, which contains 34 classes, 338 subclasses, over 1,200 supertypes, and over 5,000 individual cell clusters. And this is a lot of data to sort through when you're looking at over 4 million cells. And so a way, again, to analyze and visualize this data is by using UMaps. And as I mentioned in the previous slides, in a UMap, the dot represents individual cells, and then dots that are closer together are more genetically related, whereas dots that are further away from each other are less genetically related. And you can color the UMap by different categories. So on the first one, our graph here is showing coloring by class. So the light green cells are one class of cells, whereas our hot pink cells are our excitatory cells. Our light green cells next to them are layer B excitatory cells, and these are different classes of cells. Then our next Our next division are subclasses, so subclasses within the class. And there are too many subclasses here to list on the diagram. But as you can see here, they are color-coded and they are grouped together based on their subclasses. Another way to analyze these data is by looking at the different regions that they came from. So this is from the whole mouse brain. So we have cells from our isocortex are in dark green, cells in the hippocampal formation are light green, and the dark green and light green are close by each other. And then we have cells in the midbrain in purple and cells in the cerebellum in orange on the other side of the UMAP, because those are genetically different from cells in the isocortex in the hippocampus. We can also look at our cells by neurotransmitter type. So here we have our glutamatergic or excitatory cells in blue, and we have our GABAergic cells listed here and clustered together in red. And the data that was produced from these experiments was published in over 10 papers recently, but it's a lot of data to sort through yourself. And so because of that, we created the Allen Brain Cell Atlas, which is another online interactive tool to sort through these data and to make it more user-friendly. And we'll specifically have a webinar focused just on the Allen Brain Cell Atlas on May 22nd. And by using the Allen Brain Cell Atlas, you can look through the data from the 4 million cells. You can look at both the spatial transcriptomic data side by side with the UMAP and the single cell RNA-seq data. You can filter and look for cells that just express a certain gene of interest or even 2 genes of interest. You can isolate your cells and filter by brain region. You can filter by neurotransmitter type, and you can really dive deep into these data without having to do all of the analysis yourself. And so why is it important to define standard cell types to begin with? Another reason is that when people are in different labs and in different research institutes are studying different things, they might come up with similar results, but it's very hard to compare these studies side by side. So from these 3 studies, these are 3 studies done from different labs, and all 3 of these studies noticed that there were changes in microglia during Alzheimer's disease. However, right now, it's not easy to see if the same microglial populations are changing in these 3 different studies or if they're different microglial populations, because right now we do not have a standard definition of microglia group 1, microglia group 2, and so on. And so if by doing all this work, if we can come up with standard cell type names for our different cell types, then we will be able to compare our results from different experiments more easily and more accurately. And so the Allen Institute also created another tool which helps address this issue. So Map My Cells is a tool where if you have a cell by gene matrix, if you have your own single cell or single nucleus RNA-seq data, you can actually upload it to Map My Cells and then map it onto to a whole mouse brain taxonomy from the Allen Institute. And so here you'll get a direct comparison between your work and the cell groups and cell types that you're identifying, and you'll be able to map it onto a large, complete reference atlas of the whole mouse brain, which will give you a better idea of the populations that you're studying and what's going on in your work. And so Map My Cells, again, we'll be covering that in much more detail on April 24th. On May 22nd, we'll be going over the ABC Atlas. And February 28th and 27th are the next ones where we will be covering the techniques behind the Cell Type Knowledge Explorer. And January 31st is where we'll be going more in depth onto what is a taxonomy and how to generate them.
So thanks, Rachel, for that. Before we go into questions and let Rachel get some water or some coffee here, I just want to do a quick call out for our survey. As Rachel mentioned, we have multiple of these webinars. And so your feedback on if this content was good level, if it was well presented is really helpful for us. It's very quick, less than 2 minutes for 5 questions on that. I'll leave that up while we start going through some of the questions that were posted in the chat. And so one of them was, are there any benefits of using single cell ATAC sequence instead of single cell RNA sequence for cell definition?
Yeah, so some of the data that I showed in a few of the studies, specifically looking at the cell type knowledge explorer data. Some of that does include single cell attack data, which can show us different epigenetic signatures of these cells as well. So a lot of our work actually combines the two, where we can look at our single cell transcriptomic data and then also look at our epigenetic data as well.
Next question. You just mentioned GABAergic cells. You could also mention the different rates of disease progression and decline of pathologic cells in Alzheimer's patients. I would like to ask, could this be able to further inspire treatment of other pathologies such as Huntington's, chorea? I mean, researching the cellular understanding disease. So do you think this could be applied for clinical treatment?
Yeah, I think a lot of the examples that I showed in Alzheimer's are specifically for Alzheimer's, but we know that GABAergic cells are also affected in a number of diseases: depression, schizophrenia, Parkinson's, and we know that different subclasses of inhibitory cells are from patient samples. We know that they are differentially affected in these groups as well. So yeah, most of the examples I showed were Alzheimer's, but I think this is very valuable to study in other diseases as well, to get a better understanding of which cell types are specifically affected, how they're affected, if they're affected early in disease or later in disease. These are also important things to study as well.
Another question is, how do you define the boundary of individual cells in the MERFISH data?
Yes, so looking at the MERFISH data, they, my understanding of the experiments were that cell segmentation was used, and a number of analyses were used to define the cell, the somas specifically for that analysis. So for the MERFISH data, yes, there are things that are missing in that you're not seeing the axonal projections or dendritic arborizations of the cells. What we're really focusing on is cells segmented based on the cell bodies.
Another question is, what references do you recommend that provide specific genome markers to determine different cell types and subtypes?
Yeah, so a lot of the work, a lot of previous literature was analyzed and assessed to come up with these different markers. So historically, we know that PV and SST are good cells for genetic markers, but we're also still coming up with learning new molecular markers as well. So I mentioned one of the slides that ID2 has recently been identified as a more novel cell marker as well. And you can get this information, yeah, by looking through previous literature or sorting through the RNA-seq data that's being generated. Because this is the first time that we have a whole brain taxonomy. And so when more experiments are done and we have more brains analyzed, perhaps there are things that will change and adapt or become more accurate as we do more of these experiments. But right now, looking at previous literature and analyzing the data we have is one good example.
Next question is, for single cell RNA sequence, what is the advantages of using cells or nuclei?
That is a good question. I'm going to think about this one for a minute. One example is for humans, for human research specifically, nuclei work, single nucleus RNA-seq works in humans, whereas single cell does not. And cells give you more, doing single cell gives you more information about the individual cells compared to nuclei, and you have less bias when you do single nuclei RNA-seq. So those are a few differences between the two.
Does the Allen Institute have any web-based tool for clustering based on circuitomes, example cytokines?
So I know that in our, cytokines was the example.
Yeah.
Yeah, I know that we have some non-neuronal data in our reference taxonomy, specifically on MapMy cells, but I do not believe that contains information from cytokines. So currently, we do not have a web application for that.
Does the size of the data sets limit the addition to a temporal variable? of a temporal variable. I'm not quite sure.
So looking at, I assume this question means looking at perhaps progression of disease or at different states. So like for the Alzheimer's data sets that we have from different age ranges, different progressions, different samples throughout the disease progression and also age of our patients. And so that's a good way to look at changes, the temporal changes in disease. For our mouse whole brain atlas right now, that is just from the Allen Institute data set is from one mouse. So that's one, I don't remember the exact age of the mouse, but that's one young adult mouse. But I assume if you want to look at temporal changes, then yeah, you'd have to generate data sets based on different developmental time points as well.
Addition to that, I know there is some of the community has been asking for more different time points for the whole mouse data. And if you go in the ABC Atlas, you can actually do feedback. And if the community is making it very clear, like we would love more different stages, that will be changing our roadmap on that. So hopefully they'll eventually be coming. It's on the plans, but it changes on what our priorities are. And hearing from you guys helps change that. Is there any plans to combine all these data from a single cell RNA sequence data, nucleolide, and the MERFISH soma with proteomics?
As far as, yeah, I'm not aware of any proteomic plans soon.
For performing single cell RNA sequence data, we first need to sort the cells and then perform RNA sequence then perform RNA-seq. Doesn't the markers we select for sorting the cells affect our studies subsequently?
Yeah, it definitely can. If you look through, I mean, it depends on what the question that you're trying to answer. Like if you're specifically looking at astrocytes, then you might use a marker that specifically labels for astrocytes. Or if you're specifically looking at only inhibitory cells, you would use different markers for that. And so if you want to bias your sampling towards glial cells or towards excitatory cells or towards microglia, then it would make sense to try to adjust your sample and what you're selecting for. And so like for humans, specifically, there are a lot of glial cells in humans. And so some of our experiments, we try to down sample the glia in order to focus specifically more on the neurons. But the analysis still goes through everything and tries to capture the information from all the cells. But depending on, the type of question you're answering, then you'd want to bias, not bias, but change, be more specific in the markers that you're looking for.
And then I'm going to rephrase this question. So with the single markers for subtypes, how do you just basically determine gene markers because it will require unique combinations of markers? I know we'll talk about this more in our next webinar, but if Rachel wants to add anything about this, about how we determine a gene marker identifies the subtype.
Yeah, so when you have a subclass of cells, so for instance, you have the SST subclass of cells, and then you have cell types within that SST subclass, they have different genes that are differentially expressed within those different cell types. So some SST cells also express calbindin, some do not. Some also express protein, some do not. And so there are other genes that are used to differentiate within the different cell types within a subclass. And there are lots of different ways for defining markers. And we'll definitely go more in depth into this in the next webinar, specifically focusing on taxonomies, how are taxonomies created, and which genes do you use for selection.
And then could single nuclei isoform sequence be another layer of cell differentiation for rare subpopulations like von Economan neurons, for example? I'm not familiar with that technique.
Yeah, what was the technique called again?
Nuclei isoform sequence.
I'll have to take a minute to think about that.
Yeah, well, I'll have to get back to that one. But everyone else, yeah, no other questions. If anyone else has ones, if not, More than happy to put your questions in our community forum. If you're seeing this chat on it, thank you again, Rachel, for her time on this. And thank you guys all for attending. I'll do a last call for questions for a minute, but it looks like wrapped up. And with that. Oh, and there's been a couple questions on about cellular cerebellar cells. Yes, the whole brain includes cerebellum as well. It is actual whole brain. Do you have plans for whole brain atlas for humans? So right now they're, again, let Rachel take this. Do you have plans for whole brain atlas for humans?
Well, right now we have data from human data, specifically from we have one that's published, but we also, that's looking at, yes, we have one that's published. And then when looking at specifically our Alzheimer's data, all Alzheimer's data is specifically focused on a few different brain regions, but we have a published one, and the Allen Institute is working together with their collaborators in VICAN to come up with a newer and better one in the next few years. So keep a lookout for that.
Excellent. And then I'll answer those questions. Not, I think we're pretty much at time. So thank you again for everyone for attending. More on it on this webinar. We hope to see you for all the other webinars. Again, you must register for each webinar to receive the link.